Hello and welcome to Nikolai's Genetics Lessons, and today we are going to talk about how to read Manhattan plot. And all Manhattan plots would look similar to this picture. Manhattan plot is statistical representation method which was developed in 2000, and it was not just adopted statistical method, but it was specifically developed for genomic projects. Imagine that human genome is a massive library with 23 giant bookshelves. These are your chromosomes, and here on this picture of human karyotype, you see that each chromosome represented by two almost identical chromosomes. One we got from the mother side, another from the father side. So we have total number 23 pairs of chromosomes. And in this case, this is karyotype of the woman because we see here two large X chromosomes, and if we would see here one large and one small, this is going to be a karyotype of male. Now imagine that each book or chromosome is filled with millions of letters. A Manhattan plot is a bird's eye view of this entire library. We use it for such purpose as to find the exact typos or mutations in the books that cause things like blue eyes, height, or even diseases. Now let's take a look at the x-axis or bookshelves. The horizontal line here at the bottom shows chromosomes numbered 1 to 22 plus X and Y, but usually we do not see here X and Y chromosome for certain reasons. They're excluded. For example, Y chromosome is very small chromosome, and all genes, their functions is already known. And X chromosome, though it is large chromosome, usually we work with this chromosome separately also for certain reasons, because in the pairs of two X chromosomes, you know that one chromosome is going to be deactivated, but again, not the whole chromosome, but most of it is going to be deactivated, but still some genes still are going to be active. This means that for our purposes, we have to use different algorithm in order to analyze this chromosome. So we excluded usually, though sometimes you can see also X chromosome here is present as number 23. But if you count, we have here 22 differently colored columns, and each of them represent one chromosome. And in this case, we have 22 autosomes. On this graph, you also can see a lot of dots here, and each dot here represent single nucleotide polymorphism or SNP. Where you see solid color, it's actually represent multiple dots, but these dots are so dense here that they would be visualized as single solid color. Again, still there are many dots here. Now let's take, for example, this chromosome here. Let's say this is chromosomic pair number 7, so chromosome 7. So here it's going to be 1, 2, 3, 4, 5, 6, and 7. And here's the numbers for your convenience. Each chromosome at the top, this is where we begin counting all the genes that we can find on this chromosome. So chromosome has beginning and the end. And now let's take this chromosome here and turn it in this way. So again, this is going to be beginning of the chromosome, and this is going to be the end of the chromosome. You have to understand that the order in which genes are shown in this column is going to be the same like here. So this is going to be the chromosome, and order of genes on this chromosome, it can be hundreds and thousands of them on single chromosome. Also, order is going to be the same here in the column, but again, we do not analyze whole genes. Instead, in those genes, we have single nucleotide polymorphisms, which we analyze them here in the same order as they can be found on the chromosome. Now let's talk about y-axis. As you see, we have here p value, negative log 10, and what we see here is the strength. So here, this axis represents strength of the data. Think of y-axis as volume knob. Dots at the bottom are whispers or random noise, and we are not interested in this information at all. By the way, why this plot is called Manhattan plot? Just because you see it resembles skyline of Manhattan. So each column here looks like skyscraper. And dots here above this line is just like shouts, real genetic connections. So the whole purpose of building this Manhattan plots is to find strong relation of certain genes with certain trait of our interest. It can be, for example, height, it can be blue eyes, or it can be certain disease. Now let's talk about this line here. This is sometimes can be in red color, or as in our case, it's black. This is magic threshold. In, say, real city, you have many small buildings, but in Manhattan plot, we only care about tallest ones. And scientists draw a horizontal line, usually at the value of 7.3, or in this case it is maybe 7.4. Anything below the line is ignored. It's just background noise. And anything above the line is a winner. It means we have found a gene that is actually does something. And before I will finish today's video, I want to explain what this white gap here in the middle of the gene means. We can see it here, for example, and here. Now I want you to take a look at this chromosome. What you see here is centromere, and usually around centromere in this region there not many genes can be found. And no genes means no SNPs. So that explains this gap here and here. The centromere is made of highly repetitive DNA sequences. We also call them satellite DNA, and unlike arms of the chromosome, it contains very few actual genes. Since Manhattan plots track SNPs or SNPs to find gene disease links, there are simply fewer variations to measure in this specific region. Because the centromere is so repetitive, it is incredibly difficult for standard sequencing machines to read and map correctly. Imagine trying to assemble a puzzle where 1000 pieces are exactly the same shade of blue. That is what centromere looks like for computer. Most researchers filter out these regions because the data there is often unreliable or noisy. In genetic association studies, we look for areas where DNA is variable. The centromere is a structural anchor used for all division, for attaching to spindle fibers, and evolution keeps these areas very stable. So you don't see the same kind of skyscraper peaks of variation that you see in a gene-rich arms. In all the versions of the human reference genome, for example like HD19, these centromeric regions were often represented as N, or unknown bases, or large gaps, because we couldn't bridge the repetitive sequences yet. And this creates a literal physical empty space on the x-axis of a graph, like in this case. So this is basically all for today. I hope you now understand these graphs much better. This is going to be my first video on this topic, and more videos about how to read Manhattan plot is going to follow. And this is all for today. Subscribe and see you in the next video. Goodbye.